Well, greetings, students. Good to be back with you again. This is Dr. Jim Mitroka, and today we're going to be talking about drug discovery, development, and marketing as part of the healthcare delivery course. Just want to share with you, before we begin, my thought for the day. Finally, brothers, whatever is true, honorable, pure, lovely, commendable, excellent, worthy of praise, think about these things. And I know, as I recall my days at Bristol Myers Squibb, when it started out as Squibb, it was really an honor to work there because the founder, Dr. Squibb, was a Quaker and a man of deep faith and moral conviction. So before we really jump into the nitty-gritty of it, I just wanted to give you some idea of how things were before, let's say, the turn of the century, in the early 1900s and 1800s. Basically, anybody could sell anything and call it a drug or call it a medicine. And these products were called patent medicines. And the reason they were called that is because the ingredients were not, disclosed in the label, because the vendor would say, "Oh, this is a patent product. So we have the magic ingredients that you really don't need to know about." And this is kind of an example. This Hamlin's Wizard Oil. And then here's another interesting example where they actually do tell you what's in it, because people realize that derivatives of opium had very potent pharmacological properties. primarily sedative kind of effects, but laudanum was used for, as it says, I like it here, soothes all types of ailments. And it was used primarily for stomach upset and intestinal motility issues. But a lot of people became addicted to opium that was in laudanum. And there were really virtually no regulations at that time. You could kind of see the need for it. Interestingly enough, to some extent, we've kind of gotten back to this freewheeling market for non-prescription drugs these days. I'm sure you've seen the commercials on TV. We'll touch on that later. Well, there were two important events that led to regulating this Wild West of drug marketing. And the first of these is the elixir of sulfonilamide tragedy. And basically what happened here was that sulfonilamide had been on the market as a very safe and effective drug, really a remarkable new drug for the treatment of various types of infectious disease. But it was difficult to get children to swallow the tablets. So it was thought to market the drug in a solution form. So believe it or not, the solvent that was chosen was diethylene glycol, or now we know it as antifreeze. And at that time, there were no laws regarding safety or efficacy or anything to deal with drugs approval. And what happened was 107 people, primarily children, died taking this elixir of sulfonilamide. Again, not because the sulfonilamide was dangerous, but because of the solvent diethylene glycol. And it was a tragedy that really was of such magnitude that it called forth regulations. So in 1938, the Food, Drug, and Cosmetic Act was passed, which really was the beginning of the modern FDA. And it regulated drugs and required that you show the drug is safe. It also required that you show the drug is pure, and you had to indicate what the major components of the product were. You'll notice something here. There was no requirement back then for efficacy. Just had to show the drug product was safe. The second event that led to our modern FDA was the so-called thalidomide crisis, which is really meaningful to me because I was born around that time frame, and by the grace of God, was not exposed to thalidomide. But thalidomide, when it was originally developed in Germany, was hailed as a wonder drug for sleeplessness. So it really was great as a sleep aid. And women who were pregnant began taking it in Europe, and were finding that it really helped with morning sickness. So quite a number of pregnant women took it for that purpose. It was unknown at that time that thalidomide crossed the placenta and entered the fetus. And really what resulted was catastrophic results, primarily this pheochromelia, which is a defect in limb buds. So arms and legs would be short. There was a lot of disfigurement, horrible situation. Now, the interesting thing is that thalidomide was never approved for use in the U.S., and Frances Kelsey, shown here, is the woman who was actually given a medal by President Kennedy at the time for her role in seeing to it that this drug was not, she was part of the FDA. Actually, she was brand new. It's a fascinating story that the drug was not approved in the United States. And although the logic behind her decision is kind of depends on what you read, there's a lot of data to suggest that she did not approve the drug or would not approve it because of lack of adequate data in animals, showing that animals that are given the drug are actually exposed to the drug, because rats that were dosed with the drug actually had no effect at all, which, when you think of it, is kind of unusual, right? It's the drug that's supposed to be a sedative hypnotic, and it had no effect at all at even the highest doses. And it was later found that that was because the drug was not even being absorbed in those particular animals. And later on, additional animals like rabbits were added to the toxicity battery to ensure that there'd be a species that would emulate human beings. Fascinating story. I'd like to talk about it more, but you should certainly, as a pharmacist, you should be aware of the sulfanilamide elixir crisis and the thalidomide, probably more so the thalidomide crisis. You should be aware of those situations. All right, so then we have the modern FDA, which really was brought in the third component here, purity, safety, and efficacy. Does the drug work? Was brought into play in 1962 by the Kefauver-Harris Amendment. Unbelievable that it's fairly, recently that, just a little more than 50 years ago, that you had approved these three things. And I want you to really keep these in mind, okay? This is really critical, that you understand that purity, safety, and efficacy are what, even today, are the primary requirements of a new drug to be marketed. And notice what is not on there, okay? There's no mechanism of action, all right? Okay, let's just leave it at that. So purity, safety, efficacy. So let's take a look at some of the responsibilities of the modern FDA. Well, one is the approval of all new drug products, whether it be a prescription or over-the-counter, and that's based, again, on purity, safety, and efficacy. And then approval of generics and biosimilars. Regulation of the labeling, what an advertisement can actually say about the drug, for most particularly the indication. Then there's the issue of how the drug is manufactured, and drugs have to be manufactured according to this very specific set of practices known as good manufacturing practices. And of course, after the drug is marketed, post-marketing surveillance of a drug in terms of any kind of untoward effects or deaths or sicknesses is all something that the FDA monitors. Now, a little bit outside of the realm of prescription and OTC drugs is this issue of dietary supplements, and I just wanted to touch on it briefly here. There was a legislation passed in 1994 called the Dietary Supplement Health and Education Act, otherwise known as the DSHEA, and that was promoted to sort of obviate the concern at the time that supplements were going to be regulated and people wouldn't be able to take their supplements. The FDA, the government, was going to take it away from them. And so this law was passed. And basically what it required was really pretty minimal, just that manufacturers have to ensure that their products are safe and that their label information is truthful and not misleading. Now, I want you to take particular notice of this part here. FDA pre-market authorization is not needed. You can go ahead and have one of these dietary supplements marketed without any FDA involvement at all. Now, manufacturers can make so-called structure function claims. They can say, well, this product supports the immune system, but they can't say anything about their product, the dietary supplement, in terms of it diagnosing, treating, or curing a disease. And of course, they have to follow good manufacturing practices. in that way similar to conventional drugs. Now, here's the catch. If the FDA, and there's some crossover with the Federal Trade Commission, FTC as well, finds a product unsafe, it can remove it from the marketplace. But this is kind of, you have to be, you're assumed to be safe until proven guilty here, which can be very difficult to do. So let me give you a couple examples of supplements that were allowed to be on the market under the DSHEA and ultimately taken back. One of them is ephedra, which was used for weight loss, basically just, an alkaloid that sped up ephedrine type alkaloid that sped up metabolism. And the FDA in 2014, after finding quite a number of cardiovascular events, took it off the market. They called it a significant or unreasonable risk. Now, here's another interesting product that is on the market, Prevagen, but just to kind of give you a sense for what you can get away with under DSHEA. Okay, they get by having this structure function claim that it supports healthy brain function, sharper minds, clearer thinking. Okay, and unfortunately, they can get away with that, and they've been sued by the FDA. However, again, there's some controversy by which they manage to keep on the market. You'll notice that products, these supplements have this statement. The statement have not been evaluated by the FDA, which to some extent means it doesn't do anything. To be honest with you. Here's a summary of the timeline for drug regulations legislation, and I'll just leave that as an FYI. So let's take a look at the overall drug R&D process. What you see here is that the process can be broken up to these various areas. Drug discovery, where we're trying to select from a field of candidates, to one or perhaps a few that we're going to evaluate preclinically, in other words, in animal models, primarily for toxicity. And then we have this thing called an IND, investigational new drug, which is submitted that allows us to go into clinical trials. Now, for all practical purposes here, when I talk about clinical trials, I'm talking about studies that are done in human beings. Okay, that's a big transition going from preclinical to clinical. And then we get to a point where we've got all our human data together and animal safety data, and we file a new drug application and go into, hopefully, the marketing of the drug. The other thing I want to point out is that the transition generally from discovery to development is considered when you go from having a field of candidates to a single compound. So that's shown here from, this gray box. Occasionally, people think of going from discovery to development as being at the point of an IND submission. Either one is fine. So let's talk a little bit about how we get started with this whole paradigm of drug discovery. So what's the big idea? The big idea, and I have to point out that this is kind of a hypothetical model that I'm describing, the way that modern drug R&D is done, but many drugs have been developed by just finding things in nature and by accident, so to speak. However, this is kind of the systematic. The first way is to determine the disease to be treated and the underlying pathology. What is it that is going wrong that we can, at least in principle, fix with a compound? And we do that by evaluating the pathophysiology to come up with a hypothesis for a mechanism of treatment. And based on that hypothesis, try to identify a specific biological target that can be questioned, that can be evaluated for its activity against a field of drug candidates. So drug targets are molecular structures that when they interact with a potential drug, they can produce a beneficial effect. They're essentially a surrogate for the pathophysiological process we're trying to interfere with. And examples can be enzymes, receptors, transporters, typically. Studies with targets generally are subcellular systems, again, like enzymes, receptors, or transporters. They can be whole cells, and very occasionally tissues or animal models. Generally not, because you can't screen through a lot of animals with many lead compounds. Now, the other thing is that it's very important to establish a relationship between the target and the potential indication, that the thing that you're trying to develop the drug against. For example, if you're trying to develop a drug for cancer and you're inhibiting a particular enzyme, you want to really be able to validate that the inhibiting the enzyme can in fact reduce tumor burden, for example. So what is done initially in drug discovery is to identify a handful of what we call lead compounds. And this is done by looking at the potential activity of a great number of compounds against enzyme activity or receptor binding, for example. And typically this process is called high throughput screening. You're looking at hundreds, thousands potentially of compounds that can be of synthetic origin or natural origin to see how well they interact with the target. Now, alternatively, just to be clear, a lead compound can be identified by knowing that a particular source, perhaps a plant, has a particular pharmacological action on it. It might be that a particular plant is used by some population of people to treat skin inflammation, and then you can go that way. But again, I'm trying to just kind of go with the modern so-called paradigm of lead development, which typically uses a process known as high throughput screening. Now that we've identified a small group of compounds that have some activity against our target, we want to select from among those a particular compound that we want to carry further into animal testing. Okay, and that transition might be called into development. We're going to go from a few compounds to one compound. And how do we do that? Well, the leads are tested in vitro, quite often with human tissue, and in animal models to determine things such as the selectivity. For example, in a case of a cancer drug, it might be toxic towards cancer cells, but we have to make sure it's not toxic towards normal cells. Or in a case of a beta-1 blocker, you want to make sure that it's not affecting beta-2 receptors. That would be called an off-target effect. Physicochemical properties, what's the water solubility, what's the stability of the compound? And then, of course, metabolic properties, what's the half-life? Well, half-life is typical, or what are the biotransformation products? So from that small group of lead compounds, we select a best molecule to bring into toxicology studies in whole animal systems, and that compound is called the drug candidate. So now let's move from drug discovery, where we're looking amongst the field of candidates, into drug development, where we're taking one particular compound forward. Typically, when we go from that, we make that transition, the companies have a drug development project team. As you might imagine, in my role at Bristol Myers, I was the ADME specialist on the team. And there are quite a number of different functions, bioanalytical, process chemists, that is people, and that's really very important, people who scale up to synthesis so they can make the compound reliably. Formulation specialists, and I'm sure that you know about that from Dr. Norno. I mentioned ADME, pharmacologist, toxicologist, regulatory affairs specialist. So here's my question for you. Which of these roles could be filled by a PharmD? And I suppose in principle any of them could, but typically in this particular group, regulatory affairs specialists tend to be people who would most likely be PharmDs. So then once the drug candidate has been shown to have suitable activity, we have to evaluate certain characteristics before we can go into human beings dosing people with it. And the first and foremost thing that has to be evaluated is its toxicity in animals, so that we can have some idea of what's going to happen when it goes into human beings, what needs to be looked at. And so typically that's done by dosing at increasing levels and understanding what kind of toxicity there could be. For example, let's say there are changes in heart rate. Okay, that's something then our blood pressure could be monitored in human beings when the drugs is given to people. And then a very important thing is also the highest no adverse effect dose. Okay, so if you give 1 milligram, you find no problem. 10 milligrams, you find you're starting to see some toxicity, and 100 you see more severe. The 1 milligram where you saw nothing was the highest no adverse effect dose. Also, what is very important is to monitor blood levels and show that the drug blood levels are increasing with the dose. Because you'll recall when we talked about thalidomide, that was not the case. So if you're not seeing increasing blood levels of drug, it's going in one end and out the other, so to speak, and you really aren't going to find a dose. You're really not characterizing the toxicity in any meaningful way. Okay, so that's very important these days. And then outside of the toxicity, in separate studies, ADME studies in animals are done to determine to some extent, the absorption, distribution, metabolism, excretion of the drug. Now, I do want to mention that one can argue that the animals are not humans and that evaluating these things is worthless. But it's not worthless because, generally speaking, at least in terms of toxicity, you can get some idea of what kind of toxicity you could expect in humans from the animals. And it's very important to be able to kind of monitor those toxicities in humans in the clinical studies. So let's take a look at what document is called an IND, Investigational New Drug Application. Before you can actually start dosing human beings, you have to go to the FDA with this document, and it's a request to allow dosing people with this experimental drug. Now, this is an ongoing thing. It is submitted to the FDA at a point in time, but once the dosing does start in humans, it's followed through the whole process. They don't just say, okay, you've got an IND filed, we're not going to be dealing with you at all until you bring the drug to us for approval for an NDA. The IND is kind of a rolling document that continues during the process, the drug development process. And this is an interesting little side note that I want you to be aware of. Clinical studies can begin 30 days after the submission of the IND unless the FDA says no, okay, unless they put it on clinical hold. So it's kind of an unusual situation there that you don't actually get explicit approval to proceed in the case of an IND. If you don't hear from the FDA that you can't, you can assume you can go on. So what are some clinical study requirements? You can't just willy-nilly begin dosing people with the drug even if you have the safety data. So the dosing must be done according to good clinical practices, which include the study has to be done under a controlled set of circumstances, which is defined by a document called a protocol. That study protocol is critical, okay? And there is an individual called the study director who has ultimate responsibility for the study protocol, typically an MD. Okay? The protocol will include the dosing, safety precautions, intended use, the plans for statistical analysis, inclusion, exclusion criteria, many factors. And the plan must include a agreement document, which is called an informed consent, so that a person who's going to be involved in this study gets a document that gives, in layman's terms, what toxicities they can expect, what adverse effects. It explains that they can get out of the study anytime they want to, that they're not coerced into it. Very important document, informed consent. And then each study, each protocol has to be approved by an institutional review board. So if you're going to do a study at a hospital, a clinical study at a hospital, that there's a board of people associated with that hospital that have to review a document and say yes or no to proceeding with the study. That's independent of the FDA, by the way. So that would typically include, for example, some people who are scientists and people who are lay people, quite often even a clergy person, but individuals who can look at that study and say, I'm just not comfortable with this. For example, when I was at Bristol, we had a study protocol that required to tap cerebrospinal fluid in order to look at A beta formation for a drug that was being developed for Alzheimer's disease. And there was some pushback from the IRB because they didn't like the idea of doing such an invasive procedure. However, it was explained to them that that was necessary in order to evaluate the potential for this drug to work. And so sometimes there is a little back and forth, but ultimately it's to ensure that there's a safe and ethical administration of the drug. So 30 days after the IND has been filed, if the FDA doesn't come back with a clinical hold or a no-go, we can proceed with phase 1 clinical trials. So phase 1 studies focus on safety, tolerability, and PK properties rather than efficacy, rather than just does the drug work. So the patient population is healthy volunteers. Okay, I want you to keep that in mind. That's really the working definition of a phase 1 clinical trial. If there is a study done in healthy volunteers, even later on, it's often considered a phase 1 study. Now, note that traditionally men and not women who could become pregnant are involved in these studies. And just quickly, that is because men form sperm continuously, whereas women are born with the eggs, believe it or not, that they're going to have throughout their lifetime. So a woman dosed with a drug, if you damage the eggs, that's it. They're going to be compromised in terms of the health of an embryo anywhere down the line, whereas men, that's not so much an issue. This is really a political hot potato, but having worked in this area, I can say that it's not, at least from my point of view, a matter of discrimination. There's a very good logical, physiological rationale behind it. Now, typically these studies are not placebo-controlled or randomized. Now, sometimes they are placebo-controlled, but generally they're not. And the starting dose, so these phase 1 studies would be done at ascending doses to different small groups of people, but the starting dose is typically a fraction of the highest no-effect dose seen in animals. Remember, we talked about that. Okay, the highest no-effect dose given in animals. That's a good place to start. And the studies generally include single dose studies, and they generally include a radiolabel study to determine the mass balance or disposition of the drug in humans. Now, as I mentioned or alluded to, drug interaction studies, which can occur later in the development program, generally do. Drug interaction studies typically involve normal volunteers, so they're classified as phase 1 studies, even though they can occur later. That's kind of an oddball situation, but just be aware of it. Okay, so let's assume that we have our phase 1 studies, and basically the drug looks fairly safe and has a reasonable half-life. If you're dosing once a day, it has a half-life somewhere maybe 2 to 6 hours, 2 to 8 hours, something like that. And then we move into phase 2 clinical trials. Now the focus here is to determine efficacy and also the safety of the drug. And now we're going to be looking at patients, okay, as opposed to just healthy volunteers, and not specifically patients who have the condition that we intend to treat. And these studies are done in such a way that you give different doses to different groups of people in order to get some idea what would be the appropriate dose to use in phase 3 studies. Doses that are high enough to give you efficacy, but low enough not to have toxicity. Generally, these studies are run as double-blind, randomized, placebo-controlled, or active-controlled protocols. Okay, so let's say everything works out well in your phase 2 studies and you see some real effect. Okay, if it's, say, Alzheimer's disease, you see that there's a real relative improvement in memory compared to a placebo control group. And by the way, that hasn't happened with Alzheimer's so far. Still no drugs out there for that particular drug, but that's the main idea. So if you see a pretty good, say, efficacy safety profile, you move on to phase 3 studies. So phase 3 studies are really very much the same as phase 1 studies, but on a much, much larger scale. And now we're not looking at different doses. We've decided on the dose from the phase 2 study. And these can involve thousands of people in multiple sites. What I mean by sites is there might be different. The actual sponsor, the person, the company that's bringing forth the drug is not doing the testing in their own facility. They're farming out the protocols and the study drug to hospitals and sites where they actually conduct the studies, where they have the patients. And the results form the basis for a risk profile, risk benefits analysis. So what I mean by that is, for example, if you have something that's for a cancer indication, obviously it'll tolerate a much higher risk or toxicity than for, say, something that is for weight loss, which the risk benefits it has to be squeaky clean for that. These studies are obviously run as your traditional double-blind randomized placebo-controlled. You know that's the story. And generally, you need to have 2 convincing so-called pivotal trials that have solid statistics in order to file it. Well, in order to have an NDA approved, you can file it with whatever date you want. But in order to have a reasonable expectation of getting your drug on the market, you have to do 2 separate phase 3 studies. Huge, huge investment. This is where the billions come in. Now, phase 4 clinical trials are a little bit of an oddball here. These are post-marketing studies, so this is after the drug is approved. And really, there are several objectives for this, but one is just to get an idea of a better idea of the risk of the drug. And sometimes the FDA will approve a drug, approve an NDA, and then ask for post-marketing studies. It gets a little tricky because sometimes the sponsor doesn't do them, and there's not really a whole lot the FDA can do about it at that point. But the other reason that quite often these studies are done is to gain additional indications. There might be something in a phase 1 study that's observed. For example, it's a blood pressure medication, and people seem to be growing hair more, and you say, okay, that looks like a potential indication. Then you do a phase 4 study with the drug whose safety has already been established, and you can say, oh yeah, look at that, it really does. When we do a statistically sound study, a powered study to look at hair growth, you find that it does in fact increase hair growth. You can market it for that indication. Okay, so these again can be sponsored they can be initiated by the sponsor, for example, for additional indications, or by the FDA. Now, even though the FDA can ask for it as a requirement for drug approval, obviously the drug approval comes before the studies. So it gets a little tricky there. Now, later in the development program, along the lines of when the phase 2 or phase 3 studies are going on, if the drug is looking good, has a good benefits risk profile, other non-clinical studies are initiated that are required to file an NDA. And these are non-clinical, I mean studies that are not done in human beings, they're actually done in animals, but yet they're post-IND, okay? So these are, I know it's a little confusing, but non-clinical studies that really can't be considered preclinical. They're done after the clinical studies have started. Well, so what are they? One would be teratogenicity. Teratogenicity is just the potential for the drug to cause birth defects. Okay, and you remember our friend thalidomide there was a drug that had that problem. And actually, as I mentioned, it wasn't detected in rats or mice that were the standard models at the time, but it was detected in rabbits, ostensibly because rabbits do absorb the drug. So later on, actually, rabbits were added, as I understand, as a consequence of thalidomide. But now rabbits and rats are typically evaluated for teratogenicity in 2 species. In addition, excretion in milk. What's the potential for the drug to get into milk? And this is done with lactating rats, out of concern that a woman who is nursing, would it be all right if she's taking the drug or not? Generally, if it's not excreted in milk, you don't have to worry about any issues with the baby. And conversely, you would have to worry if it was excreted in milk. And maybe it would be contraindicated in a nursing mother. The third non-clinical test battery that's often done is these 2-year carcinogenicity studies. Now, obviously, this would be for a drug that's going to be used for a long time. If it was an anti-infective just to be given once or twice, it wouldn't be. But for, say, a drug for blood pressure or type 2 diabetes, you would have to do a 2-year carc study. Now, again, just to put this in context, these are animal studies, non-clinical studies that are done post-IND. You might say, well, why are they done post-IND? They are very expensive and time-consuming. So a sponsor is not going to initiate these studies if the drug bombs out, if the drug doesn't have real potential for filing. Okay, so they're non-clinical studies that are done post-IND. So this slide, which is taken from the textbook, just kind of summarizes pretty much most of what I've been talking about at this point. You don't have to really know the details, but just kind of have an idea of the general features. So then when all the data is put together, the early discovery data, which would be like against an enzyme target perhaps, and then the preclinical, the non-clinical, the animal data, which is mostly toxicology, when that's all done, and then you have your clinical studies, which the focus, of course, being on the phase 2 pivotal studies. When all that is accumulated, and the sponsor feels as though it warrants going further and marketing the drug, all the information is put together in a new drug application for conventional small molecules, or what's called a biologics license application, BLA, for biologics. So this, in the pre-digital transformation age, involved literally truckloads of documentation. Was just unbelievable amount of documentation, and it still does involve a huge amount of documentation, but it just happens to be digital now. So that's all shipped to the FDA for their review. And by the way, that's another potential job for people who are PharmDs, is to be an FDA reviewer. So what's in it? Again, I don't expect you to memorize any of this, but you have to show, obviously, we've already been talking about it, data to support the safe and effective use of the drug, that the benefits outweigh the risk, that the proposed labeling is appropriate. That's what's stated on the package insert and on the actual physical label on the bottle is appropriate. That is really a critical component. The wording has to be smithed just right for that. Then the last thing I have here, again, which is important, manufacturing methods and controls. You have to show that the identity, strength, quality, and purity are consistent with that product, which is one of the reasons that drugs that are approved through the FDA are different than these herbals. A herbal may be effective, but from batch to batch, there could be tremendous variation. You don't know what the shelf life is. This all has to be really proven and established for an NDA or a BLA to be approved. Now, a standard review takes about 10 months. Well, let's call it a year. Okay. Where an expedited review, okay, and we're going to talk about how those come along, might take about half that amount of time. So the FDA is sitting on this for quite a while, and it's because there's just such a huge amount of information. By the way, nowadays there are what are called user fees. The sponsors generally have to, well, they don't have to, but they volunteer to pay a certain amount of money to have the NDA or the BLA reviewed by the FDA. So these are the different components of an NDA: medical, biopharmaceutical, chemistry, microbiology, statistics. Really, I'm just listing them here, FYI. But again, there's a lot that goes into it. There are these formal sections of the NDA, and essentially you have a regulatory affairs person who is the go-between between the sponsor and the NDA, and make sure that all this is put together in a clear, meaningful way. So remember, the objective of the FDA is to assure the drugs are safe, effective, and pure. That is just fundamental as a pharmacist. You should know that hands down. Okay, just a summary of FDA expedited review programs. I am not going to really go over these individual programs. These are all fast track, breakthrough, priority review, accelerated approval. These are all expedited review programs that the FDA has. And each one has kind of its own subtleties about the requirements. But basically, in a nutshell, the drug, if you want a drug to undergo expedited review, either it has to be for a unmet medical need. For example, Alzheimer's. If you had some drug that was shown in phase 2 studies to be effective for Alzheimer's, you know that's going to get expedited review. Or if it's something that is a breakthrough, like meaning that it's so much better than other drugs that are available for the indication. For instance, if there was a new cancer drug that really improved lifespan, but was not had a very, very low toxicity, that would be something that would get expedited review. I will say, though, that it's really amazing how the pharmas can be creative in suggesting that there's something about their drug which warrants expedited review. It could be like, well, ours doesn't have this drug-drug interaction that the other drug on the market does. And it really can be a judgment call whether or not a new drug warrants expedited review. The other sort of non-standard filing review process involves the Orphan Drug Act. Now, the Orphan Drug Act was passed by Congress to allow drugs to be marketed and developed that are for relatively rare diseases, and it's kind of arbitrarily set at less than 200,000 patients. Examples would be Huntington's, ALS, Tourette's, muscular dystrophy. And the drug development process and requirements are essentially the same, except it does not require large-scale phase 3 studies. Okay, so again, keep this in mind. as compared to expedited review. Expedited review is a process that exists for drugs that are out to the general public, but they represent some sort of a breakthrough. Okay, so they get an accelerated review of the NDA. Here with the Orphan Drug Act, pretty much the review is the same amount of time, but it's just that the number of patients involved in the large studies is less. I mean, that kind of seems like it's a logical consequence of the indication here. And the objective of the Orphan Drug Act really is to make it financially worthwhile for companies to even investigate new drugs for these disorders. And this gives you just kind of a list of some of these drugs, and it's surprising, really, that rituximab, such an important drug, for example, is considered an orphan drug, but it does apparently it meets that under 200,000 patients criteria. So the FDA's role doesn't stop once a drug is approved to be marketed, once an NDA is approved. And there are two specific areas that they look at. One is safety. Not all adverse effects can be detected with clinical studies. If it's one in, say, 1 in 5,000, and you do a study in 10,000 patients, you might not even see it, or you may get 1 or 2 and just think it's statistical fluke. Okay, so the FDA follows up for safety even when the drug goes in the general public. Like Vioxx is an example of a drug that once it was in the general market and millions of people were taking it, there were issues with its safety that became clear. But just so you understand as a pharmacist, not just because a drug is authorized for marketing by the FDA doesn't absolutely mean it's not without risks. The other area that FDA keeps an eye on is the manufacturer of a drug, because you may recall when we were speaking earlier about requirements for an NDA, you have these manufacturing requirements for purity. And so later on, post-marketing, it might be that the manufacturer begins to get the drug from somewhere else. They could out-license the manufacturer to China or whatever. But the FDA periodically inspects the facility to make sure that it's being made according to specs. Very important aspect of their work. So let's talk a little bit about intellectual property rights. It costs a lot for sponsors to develop a new drug, and they want to be sure that they can recoup their investment, because you have to remember that it's not only that you're paying for the development of that drug, you're paying for all the drugs that failed, all right, that have to be incorporated into those costs. So the intellectual property, well, I've gone over a lot of this, what it's for. But the main thing is it provides a period of market exclusivity. That's really what we mean here by intellectual property. Marketing exclusivity. No one else can sell the drug that you've put all that money to bring into the market. And there's really two components to this that you have to keep in mind. One is the patent, okay? The drug has a patent as a new invention, so to speak. And then there's these additional periods of exclusivity that are given by the FDA under certain conditions. We're going to get. So as I mentioned, as with other types of new inventions, one gets a patent from the patent office for 20 years. And here's the thing. The drug has to be a new chemical entity in order to get that patent. So it is true when somebody will say, well, here's something that is a product which we see every day from nature, and no one is developing it as a drug. To an extent, it's true, unless they can come up with something unique, maybe a unique formulation. that you can't get a patent on the material if it's not a new chemical entity in terms of drug development. These patents are issued by the patent office. So the interesting thing is that since the patent runs for 20 years, typically a company will file for a patent on a lead compound early in a discovery to protect rivals from developing that compound. Okay. The downside is you got to remember it takes about 15 years for a drug to go from inception to NDA, which means that, wow, 3/4 of your patent life has already been chewed up just in the discovery and development phase. So it's kind of a tricky thing to when a company should actually file for a patent on a new drug. They want to start early so nobody else can get that drug, start to develop that NCE. But on the other hand, they want to wait a while so that they're not, they have more exclusivity once the drug is filed. Now that's strictly in reference. So beyond the patent rights, the FDA also gives the sponsors exclusivity that starts once the NDA or BLA is approved. Again, these are sole marketing rights, which are independent, let's say, of the patent rights. And it depends on the nature of the compound that is approved. For example, an orphan drug, remember that's for a drug that's for a relatively small population of people, gets 7 years post-approval exclusivity, whereas a traditional NCE would get 5 years. And if a company does develop a non-new chemical entity drug, if they take the time to do that, they get 3 years exclusivity. Interestingly, if you do the studies that are necessary in children, you get a pediatric exclusivity of 6 months added to the former exclusivity here. And the other thing I want to mention is biologics are interesting because they're extremely difficult to develop. So the FDA has decided to give biologics 12 years of exclusivity beyond the approval date. Now this is a little tricky, I know, but the easiest way to think of it is that a generic form of a drug, okay, because the generic houses are just waiting until the exclusivity periods expire so they can market basically the same drug for a discount price. The generic form cannot be marketed until both the patent and the exclusivity period have expired. So this slide kind of brings it together a little bit, and let's look at the case of a new molecular entity that had a patent for 20 years, and maybe that was filed just a little year after the drug was first synthesized. All right, so that patent goes beyond a few years beyond when the NDA is approved. The NDA might have been approved roughly 15 years after the drug was synthesized. All right. Now, also, there's these exclusivity period by virtue of the fact that the drug was approved by the FDA and as an NCA was given this period of exclusivity. Now, when can the generic manufacturer begin to market his or her drug? Well, after both those periods, the FDA granted exclusivity and the patent exclusivity expire. So in the case of our NCE here, we see that the patent goes out further. That's really what's going to determine when a person can file a a generic request. Whereas in the case of the orphan drug here, because it has a little bit longer exclusivity, that's going to be the rate limiting step for filing for a generic approval. Now the other thing is that biologics, I do want to point out, because they have this really long post-approval exclusivity, are somewhat more. That's an extra incentive to the pharmaceutical companies to try to manufacture and get those drugs to market. Let's take a look at what's required for a new molecular entity, which is called here a small drug, versus this is a new drug being brought to market by a sponsor versus a generic version of that same molecule. So you could see that in terms of the overall R&D for the new molecular entity, you have to demonstrate that the drug is safe and effective for target indication. Whereas the generic, and I want you to be clear on this, all you need to demonstrate, although it's challenging, is equivalence to the approved chemical entity. Typically that's done by showing a pharmacokinetic curve which is indistinct from the original innovator compound. Not better than, not worse than, it has to be identical PK profile to the original molecule. That is all you have to do. So if you look through the other requirements for a new molecular entity, they've got in vitro testing, non-clinical, phase 1, phase 2, phase 3. Oh, let's see, and the cost is over $1 billion. Let's see about the generic. Whoa, these guys, do they have to do clinical testing? Here we are. Only that PK study to show similarity, but no phase 2. Generally, for our purposes, we're going to say never. They don't have to do phase 3. They don't have to do non-clinical studies. None of those things are necessary, because if they show that the drug has essentially the same PK profile, you've established you're giving effectively the same thing as the originator compound. Okay, so if you have questions about that, let me know, because that's kind of really, I think that's really important, especially for you to understand as pharmacists. Okay. Now, there's an analogous situation with respect to new biologics and biosimilars, but I don't want to really get into that, because it's really very, very hazy as to how you establish that a biosimilar is similar, is effectively the same thing as a new entity biologic, but it's a kind of an analogous process. Just a word or two about direct-to-consumer advertising. The FDA does allow direct-to-consumer advertising as long as they provide certain key points of information and that they have a fair and balance between risks and benefits. Now, this is obviously in the eyes of the beholder, okay? The good thing is that it educates consumers. The bad thing is that, it ends up can cause an artificial demand. Patients running to their doctors saying, I saw this drug on TV, please give it to me. Interestingly enough, only U.S. and New Zealand permit direct-to-consumer advertising. So the ads have to they have to show at least one approved use, generic name, adequate provision of safety. That's why you often hear them talking so quickly at the end about their whole list of adverse effects. I think that the thing that often patients get a little concerned about is that there's this big list of AEs given at the end, and by regulation, anything that popped up in the clinical studies has to be included as a potential AE, and it can really sound daunting sometimes. Now, of course, the sponsors try to get away with what they can, and sometimes the ads aren't 100% faithful. And this is just an example of an ad for Xarelto that said in the original print ad, there are no dosage requirements. That was for renal impairment, which the FDA took issue with and made them include this little disclaimer in their print ad. That was for the public. The label or the package insert for the drug, as we've said, has to be very specific about the indication, the use of the drug. But there's also uses which are not specified on the label. Let's talk about that in a minute. Those are called off-label uses. Now, once a drug is approved for marketing, the FDA really has very little power to do much about what's said about that drug by the sponsor. And physicians can technically prescribe any drug that's been approved for any condition. So they could give you a chemotherapy drug for a headache, and in terms of drug regulations, that's okay. They can't be cited in any way for it. So these off-label indications, the FDA doesn't like it, though, and basically these can be found serendipitously. Again, as I said earlier, you can find that a drug for blood pressure causes hair growth, which could be good, could be bad, but that might be found serendipitously during the drug trials. And in order to really have that as what we call a labeled indication, what you have to do is additional phase 3 studies to prove that in a powered study. Here's the thing. Here's the important thing for you to keep in mind. It really is, especially as a pharmacist. No one, especially a representative of the sponsoring company, cannot promote an off-label indication of the drug. This gets a little tricky if they, exactly what we mean by they're promoting it, but technically that is the law. A physician can write out a script off-label, but a sales rep or a medical science liaison cannot promote the drug for an off-label indication. An example would be gabapentin, which was approved by the FDA for seizures. That was its labeled indication, but it was marketed by Warner Lambert, by their sales force, for psychiatric conditions, and they were sued big time, almost $500 million for that. So let's take a look at some future trends in pharmaceutical R&D. One is an expansion of biologicals. About half the filings for new drugs are BLAs, and that's expected to go to about 70% by 2025. And I do want to mention as an aside, the problem for the consumer is that the biologics are unbelievably expensive, typically over $100,000 a year for treatment, and I'm really not sure what can be done about that, but that's the case at present. Another trend is direct patient involvement in healthcare. I think you're seeing that. You'll see it in the pharmacies that people come in and they've looked up things in PubMed and Medline and just basically online, and then they're wearing their wearables that monitor heart rate and blood glucose levels. A lot of direct patient involvement instead of just relying on the healthcare professionals to dispense wisdom. There's a team-based approach to healthcare, including pharmacists, with things getting more and more complicated all the time with the, for example, my favorite area, pharmacogenetics, and physicians having a hard time keeping up with that. Other specialties will become part of a team to help make those decisions in a meaningful manner. A focus on prevention. Hopefully, 80% of chronic diseases are related in some way or another to lifestyle choice, like type 2 diabetes and coronary artery disease, for example. And then the one that's near and dear to my heart is advances in genomics. It's providing rational basis for drug targets. And of course, if a person then, let's say they have migraine, and they come in to a physician's office and want to have be evaluated for what is the appropriate drug therapy. If a drug was developed that was for a particular subset, a genetic subset of people with migraine, those individuals would have to be tested to see if they have that genetic variant. That's called companion diagnostics. And we'll see more of that. I suspect that you, as a pharmacist, will be seeing more companion diagnostics as time goes on. Well, just to kind of have a little bit of fun with the genetics part of it, 23andMe is getting serious about drug development, could signal a fresh new approach. Since this is my favorite subject, just to elaborate on it a little bit, the slide shows a genome-wide association study where we have people that have a particular disorder, they're the cases, and people who don't, they're the controls. And you match their gene variants, you can see that there are certain variations that pop out in the case population. Knowing what that genetic variant is, in principle, you'd be able to find out whatever enzyme or transporter it was associated with, develop a drug that would interact with that enzyme or transporter, and then stratify your patients so that you're just going to look at those patients who have that variant in terms of your drug potency. Now, this is very interesting because this represents an alternative to the traditional blockbuster approach that, like I was familiar with when I was at Bristol Myers Squibb. You try to develop a new HMG-CoA reductase inhibitor for everybody and hope that the overwhelming number of people, or the majority of people who have hypercholesterolemia, have that, are susceptible to that particular drug. But often that's not the case. So we're getting away from that with this paradigm, and you're saying, okay, there's just a subset of people with that disease state that we can treat. But the thing is, you're relatively certain that that patient population will respond. Okay, that's sometimes called targeted therapy. And that's already ongoing. Glaxo hooked up with 23andMe to identify these kind of genetic variants in people who have particular conditions. I know there's some issues here regarding privacy, but for the most, as far as I know, there's never been a case when any of these big providers like 23andMe or Ancestry.com have given out identified information. So let's talk a little bit about common myths with drug R&D, and I have to be careful not to get emotional about these because some of these really get on my nerves. Drugs are a ripoff. The drug costs pennies a pill and they sell for dollars a pill. What can I say? Yes, it's true. In terms of cost of goods, that's a very small component of drug cost. Most of it is because of the expense of clinical testing, the regulations, and the fact that you have to somehow pay for drugs that never actually made it to market. And I will say there is a lot of profit being made. Drug companies hide known toxic effects of drugs. I have to tell you, truthfully, when I was at Bristol, I never saw that even once. People who work for drug companies are human beings just like you or I would be, and I don't ever remember once in over 20 years being asked to compromise my Christian values in terms of hiding information. Drug testing on animals is unnecessary. Well, at least for now, there is no suitable alternative. It's limited, but it's kind of like necessary but not sufficient. Drug companies I love this one, drug companies have cures for diseases, but hide them so that they can continue to treat symptoms. I'm going to leave that one for your own imagination. Obviously, I don't think there's much truth to it. And just quickly, drug discovery is a very, very, very expensive and wasteful game. You can see that this classic drug R&D pipeline shows how many compounds are screened for a particular indication before one or two comes out the other side. And this is something that you may not be aware of. I think I alluded to it earlier, but the cost of developing new drugs has gone up exponentially. It's just unreal, and part of that is because the low-hanging fruit, so to speak, has been picked. The easy drugs to develop, that's done. Part of it is because of the additional requirements for clinical studies that there are and for drug testing. And part of it is just the regulatory burden. There's a number of issues there, but the fact of the matter is that the cost of developing new drugs has gone up tremendously. Well, okay, gang, I think that kind of wraps it up as far as just an introduction to PharmD. I think this is an interesting quote. I just want to share it with you because I know this was kind of my experience in the industry. You can read it on your own. And yeah, I want to leave you with these references. Now, usually I know students don't pay a lot of attention to references, but they're there to help if you want to go back and look at some of this info in more details. But the one I do want to particularly point out is this Industry Pharmacists Association, also called the IPHO. You may be aware, we have a chapter here at GSOP of that organization. But they have a website that has some really good information if you are even thinking about possibly moving into a career in this industry. They have firsthand accounts of PharmDs who work for the pharmaceutical industry, and they describe their role, their job, and what it took for them to get to that place. Okay, my friends, I think we're going to leave it at that for now. God bless. Just shoot me an email if you have any questions, and I'll, God willing, see you in class. Bye-bye.